Good afternoon. This is Shashikala Patyal from Hansraj Mahila Vishwavidyalaya, Jalandhar, Punjab, India, and Prahashit Sharma from Indian Institute of Information Technology, Allahabad, Uttar Pradesh, India. And our video abstract presentation for LanBix-2021, the first Sri Lankan bioinformatics conference, is on the topic correlation between coverage and N50 values, Hamming distance, all three of which are essentially dimensionless quantities towards genome assembly and compression of randomized Novaseq-6000 reads. Novaseq-6000, as we know, is the benchmarked NGS sequencer used for whole genome sequencing data generation at Center for Brain Research, Indian Institute of Science, Bangalore, the nodal coordinating institute for Genome India project sponsored by Department of Biotechnology, Government of India. So let's understand the basic terms. Perfect coverage in fragment assembly is the concept of having a read or k-mer that begins at every nucleotide. The sequencing coverage level often determines whether variant discovery can be made with a certain degree of confidence at particular base positions. N50 is a measure to describe the quality of assembled genomes that are fragmented in contigs of different length. It is the shortest contig length needed to coverage 50% of the genome, and Hamming distance is the exact number of substitutions that is required to convert one genetic string to another. So what are we aiming to do here is to essentially fundamentally interlink these three quantities in the context of genome assembly of the human genome. So essentially we start off with using the Randomized Python script developed by Morris et al. At Padana University, School of Data Sciences, Malaysia, with a fixed length of 35 base pair single end chemistry of the Novaseq-6000 and exactly 4.12 sequences, so that we have a reasonable inter-read Hamming distance between them. And we need to find out what is the exact Hamming distance at which a particular fixed length read will give a perfect coverage. And in order to convert the multi-FastA files so obtained to FastQ, we use the BBmap reformatted SS script, and with plugging in good qualities in this range of Q28 to Q40, FRET plus 33 offset. And then we use a mini ultra-fast single-end genome assembler developed by Ryan Chicky at INRA, France. And then we take the Genome Reference Consortium 38 human reference genome and systematically introduce sequencing errors and substitution errors as well as indel errors, so as to generate pseudo VCF files, which then are subject to VCF consensus Perl script, so as to generate individual alternate genomes. And then finally, we do a multiple sequence alignment of these sequences using tools like Cluster Omega, MAFT, KL, and Muzzle, MVU, TCoffee, and so on. And then we need to note that 99% of the genomic content and base composition is the same across all these genomes, and then we need to only store the differences in the genomic segments taken together, for which we use a VCD based delta differencing command line interface tool such as XDelta3 coupled with elementary cellular automata compression that yields 4% more compression in a sublinear manner similar to genetic algorithms. So that's about it, and fundamentally we want to signify that we are trying to interlink perfect coverage, genome compression, genome assembly, as well as Hamming distance and N50 values. Thank you.